Today we will be understanding inflammation. So let me write it down. Inflammation. Inflammation, that's the topic for today, and we're going to cover all the details of inflammation. First, we need to know what inflammation is. Inflammation is a protective response. Okay, it's a protective response that is initiated by our body against infectious agents, against toxic molecules. So it happens immediately after whenever there is infection. So if there is any infection, foreign invader is coming inside or trying to enter inside the body, that's when it is getting initiated. Second, tissue injury, when there is cut, there is injury to our organs. When there is toxin exposure, if accidentally you get exposed to toxins and all, our body is going to initiate the response. And then cellular stress is another factor. Whenever cell is under stress, that can also cause inflammation. Inflammation is really, really important topic. Now, what are the signs of inflammation? These are different signs that indicates inflammation. One is redness. Whenever there is redness in a particular area, which is also known as rubor, R-U-P-O-R, and that is indication of inflammation. Second one is when there is warmth, means there is high temperature, which is also known as calor. Okay. And then third stage, which is, or third property, which is swelling, whenever there is tumor, whenever there is swelling of that region. Next is pain. It's also denoted as dolor, right? So these are the words, specific words that are, mentioned in inflammation. That's why I'm mentioning over here. Next is loss of function, which is also known as functionalism. So these are the terms that are used for the inflammation. All right, now let's talk about the step number one, which is when there is entry of the foreign invaders. So let me show you by designing the illustration. I'm trying to design a human hand. Okay. Oh, oops. Okay. Yeah, we are back over here. So just imagine this is a human hand, and while you're working and doing things, you got a cut from any metallic things, maybe, right? I'm just making it up. Now you got the cut and you got the tissue injury. Now what happens is the first line of defense, which is skin, got destroyed. So that means there is this entry pathway. There is a deep breakage in the skin from where the external microbes that are present in the environment that can actually come. So through that cut, which is there in the skin, the pathogenic organisms, it can be, let's design bacteria right over here, but many things can come down, right? You can have, like, for example, this bacteria, came in because we have direct portal of entry open. Now viruses, this is the resemblance. I'm trying to make a structure of virus. You can also have parasites, small parasites entering through these cuts and wounds, right? So after these they enter inside the body. These pathogens basically can actually have molecules called PAMPs, pathogen-associated molecular patterns, and host will have PRRs. These are, pattern recognition receptors. They will bind with PAMPs. Okay, so pattern recognition receptor example is different TLRs. They are present on macrophages and also on the dendritic cells. So different TLRs are there. So these are known as pattern recognition receptor. Example is TLRs. On the other side, on the microbe side, you have PAMPs. Okay, you have different molecules. They are associated with microbes. For example, lipopolysaccharide, LPS, highly potent inflammatory molecule. Peptidoglycan, the component of the bacterial cell wall. Again, it can induce inflammation. You have DNA. And that is coming from, let's say, viruses that can also cause inflammation. You have fungal beta glucans, right? They can also cause inflammation. So there are various molecules that can cause inflammation. And this is the step one where these molecules are going to interact with other molecules. Now, you have another range of molecules called DAMPs. They can also cause inflammation. Now, what are DAMPs? DAMPs are coming from our own cells. So let me design the host cell, and let's imagine these red cells, they are the dead cells. For some reason, the cells, they are, they are dying in our body, and we have these two cells They are dead cells. The nucleus is also destroyed. Look at that. Nucleus is destroyed. Components are also leaching out, right? These are dead cells. Dead cells, they have DAMPs, DAMPs, damage-associated molecular patterns. So you have molecules that are coming from the dead cells. For example, ATP can get released from these cells, right? And that ATP can act as DAMPs. You have uric acid and you have crystals of uric acids, right? So that can also be act as DAMPs. You have HMGB1 protein that is coming from damaged nucleus. So nucleus releases this protein and it can also act as a marker that can cause inflammation. You have mitochondrial DNA. Mitochondrial DNA is usually present inside the cell. But if that DNA is detectable in the host, that clearly indicates that you have damaged cell, right? So in essential, what you have is you have nucleus, and nucleus will give you, let's say, damaged nucleus will give HMGB1 protein that is coming from nucleus. You have mitochondrial DNA that is coming out. That is also one of the indicator of inflammation. You have uric acid crystals. These are the crystals that I'm trying to make. Uric acid crystals. These are also markers, and they are known as DAMPs, right? And further, you have molecules like ATP. These are the ATP molecules that are coming from the cell. All right, now let's talk about toll-like receptors, TLRs. And these are the surface receptors, and they are also present inside the endosomes. I'm just making the receptor structure right over here. You can see you have, this is the TLR molecule present on the cell surface, right? And you have different TLRs that are present, and they detect specific PAMPs. TLR4 will detect lipopolysaccharide. You need to remember that. TLR2 will detect peptidoglycan, which is present in the cell wall of bacteria. TLR3 molecule will detect double-stranded RNA molecule. Okay, these are the ligand for these TLR. TLR9 will detect unmethylated CpG-rich DNA molecule. So there are specific ligands for specific toll-like receptor, and based on that, our immune system will work. Further, with this interaction, what is going to happen, that this interaction is going to activate MID88 dependent pathway. So there is the protein called MID88 protein that can get activated, and it is going to further activate downstream molecules, and those molecules are IRAK4. You need to remember the names of these molecules, TRAV6 and TAK1. These are the molecules, downstream molecules that can activate nuclear factor kappa B. It's a master regulator molecule. Or it can also cause the activation of MAP kinases. All these molecules, they are really important for gene expression. So let me design the molecule for MAP kinase, and next is, in bracket, I'm going to write gene expression. So it's going to cause the expression of various genes. Now let's further talk about NF-kappa B activation. How the activation will happen. So let me write it down. NF-kappa B activation, or further the expression of cytokines, because whenever there is activation of NF-kappa B, you will have cytokines that are getting produced. So what happens is, usually NF-kappa B molecule, if this is the protein, it remains bound with a molecule called IKB. It's the inhibitory molecule, and now that complex is in the inactive state. Okay, now whenever there is inflammatory signal or inflammation happens, then the second part of this, which is IKB, that gets degraded. So let me show it by making these small structures, and basically that represents the IKB is degraded, right? So IKB degradation, or IKB is degraded, and then this molecule now can move inside the nucleus. So this is, just imagine this is the nuclear membrane, and now the molecule is going inside the nucleus here, right over here. Now molecule is inside the nucleus, NF-kappa B. That in nucleus is going to start the expression of various genes called inflammatory genes, or those cytokines. Okay, so let me write it down. Inflammation or inflammatory associated molecules are going to get released right over here. So what happens is, basically NF-kappa B, that protein, let me design that protein right over here, is going to bind to the DNA, and it will have a region, a specific region where it can bind, and then it will start the expression of various molecules called cytokines. These round structures that you're seeing, these are the cytokines that are going to get released, and those are the effector molecules. And what the cytokines will do in our body, let's understand that. Cytokines, first cytokine that will be released is NF-kappa it's not NF, it is TNF-α. It is pro-inflammatory cytokine. It's going to cause the increase in inflammation. It's going to cause the increase in fever. It's going to cause vascular permeability. Very, very important. Vascular permeability will be caused by TNF-α, tumor necrosis factor alpha. Second cytokine is interleukin-1 beta. Again, it's pro-inflammatory cytokine, which is going to induce fever in our body. So these are really important cytokines that are related with fever induction in our body. If there is fever, probably these two molecules are working at the back end. You have IL-6, which activates liver to produce a protein called acute phase protein. Okay, and this also causes the formation of serum amyloid A protein. Serum, along with acute phase protein, you have serum amyloid A protein. Really, really important molecules in inflammation. Next cytokine that will be produced is IL-8. It's also known as CXCL8. Now, this molecule is important for neutrophil attraction. So this is, just imagine this is neutrophil. This is going to get attracted towards the site of infection. So neutrophil chemoattractant, I can write it down. Chemoattractant means it's a chemical molecule that can attract neutrophils to the site of infection. Okay, so neutrophil is really important when it comes to phagocytosis and other immune-related processes, but it's going to come when IL-8 is going to call our neutrophils. All right, next we have is endothelial activation. In inflammation, endothelial activation is also really, really important stage. You also have leukocyte recruitment during this particular phase. So how leukocytes, they are going to come to the site of the infection. So let me write it down, leukocyte recruitment. That is also the part of inflammation, where endothelial activation is going to happen, and then leukocytes, they are going to come to the site of infection. So the story will again start with the cytokines, TNF-α, and then you have IL-1. These cytokines, they are going to activate the endothelial cells. So with an arrow, okay, endothelial cells, especially endothelial cell lining in the blood vessel, they are going to act on those linings, right, of the blood vessel. Now, what will happen? These cells, endothelial cells, they are going to start expressing. They're going to start expressing adhesions. There are adhesion molecules that are going to get expressed. For example, one molecule which is really important, first one, let's talk about that, is P-selectin. P-selectin is really important molecule, and this molecule is again related with specific bodies that are present. Whenever there is histamine stimulation, this molecule is going to get activated through Weibel pallid bodies. These are the bodies. They are also known as Weibel, W-E-I-B-E-L, Weibel pallid bodies. And that will happen immediately after the histamine stimulation, right? Histamine stimulation is going to cause that reaction. Okay, so let me write it down, histamine stimulation. The next one, really important, is these proteins, P-selectin, they are going to cause the attachment of the cells, these leukocytes, to the blood vessel wall. So the attachment is going to happen because of this P-selectin, which is present in the Weibel pallid bodies. And the second one is E-selectin. And again, in the presence of TNF-α, IL-1, P-selectin is going to get further, synthesized. Okay, let me show it and write it down first, TNF-α, IL-1, and because of that, it's going to get synthesized, and this is going to help in rolling interaction. Okay, rolling interaction. So it's going to synthesize, and then it's going to help in rolling interaction, where the leukocyte is going to roll on the wall, and then further interaction is going to happen. That I'll show you in detail with illustration, right? But you need to know that you have P-selectin, E-selectin playing very, very important role in attachment, and also E-selectin is playing very important role in rolling interaction. Now let's move on to the next one, which is integrin activation and adhesion. Integrins are really important protein that play essential role in the attachment, and how that happens, as I told you earlier, right? There is this rolling of those cells, neutrophils or leukocytes, and then through that rolling, what happens is there is activation. So chemokines are going to get released, right? Chemokines which are present on this particular wall. So you have chemokines that are already present right over here. So these yellow colored dots, these are chemokines. And now what these chemokines will do, they will activate the integrins that are present right over here. So activates the integrins, and that will happen with the GPCRs, G protein coupled receptors, right? G protein coupled receptors. Now, what are integrins? Integrins are nothing but the surface receptors. Okay, integrins. Let me show with the illustration. Just imagine, this line is the surface, and on the surface you have receptors like that. And these are, as I told you, these are receptors. And then what happens is, whenever there is activation, these receptors, they change their conformation, and they transit from low affinity to high affinity receptors. Okay, because they are really important in adhesion, right? So after activation, what is going to happen? Low to high affinity change is going to happen. And the example, if we talk about example LFA1, which is also known as CD11A / CD18, you have MAC1, which is CD11B and CD18. So these are the really important integrins that are playing an essential role in this process. Next is, let's talk about endothelial ligands, right? Because you also have ligand molecules that are going to be there for the integrins, right? So on the endothelial cells, you have ligands for those integrins. You have ICAM, you have VCAM. So you can see the green color right over here is ICAM, the white one is, let's say, VCAM1, ICAM1. Then these are the ligands for the integrins. Basically, integrins, they are going to bind with these endothelial cell ligands. And that will cause a firm adhesion. Very, very strong adhesion will happen with the endothelial lining, and those cells are going to attach very, very strongly to those lining. What is happening? These cells, they will move from the endothelial, inside the blood vessel to the site of infection. So first they have to attach. So all this process that is happening is because they have to move through this particular wall. Now that is the movement called, the process called diapedesis, where the cells, they are moving through these very, very small spaces that are present in our blood vessel. It's also chemotaxis. Diapedesis, chemotaxis means there is a very, very small space in the blood vessel from where these cells are going to move. So the squeezing of the cell will happen. During the diapedesis, the cell is going to squeeze basically through these tiny, tiny holes, and this process is known as diapedesis. And obviously that will happen in presence of chemical molecules, right? So they're going to squeeze themselves and then move to the site of infection. And this process is known as I told you, diapedesis. It's also known as transmigration, okay? So that you understand and you remember transmigration, right? And then as you can see over here, as I told you, that chemoattractant will be released. Cells, they are going to follow these chemoattractants, right? Because concentration gradient is going to get formed. So as you can see here, you have high concentration, low concentration. So cell is going to move in this direction. Look at that. So this is how they're going to move, and then follow the chemo, attractant molecules. And these are the molecules which are essentially interleukins. You have those chemotactic molecules. They're also known as chemotactic molecules. You have interleukins, you have complement molecules. And what are those molecules? Let's talk about those also. IL-8. IL-8 is very, very important molecule. Complement C5b is in that category. LTB4, and then you have FMLP. These are the molecules that are really important in this particular process, right? So this includes our endothelial ligands, integrins, and also endothelial activation. Now let's talk about the next process. All right, now let's talk about rearrangement of the cytoskeleton, which is, let me write it down, cytoskeleton rearrangement. How the actin cytoskeleton inside the cell is going to rearrange itself so that cells can actually migrate from one place to another one, right? So let's imagine this is the cell, and you have a nice nucleus, you have other structures, and then you have actin cytoskeleton. In the green color I'm trying to show you, there is, these actin filaments that are present inside the cell. So this is the actin cytoskeleton, and in the presence of GPDAs, this entire process is going to happen. You have various molecules that are playing an important role. You have one protein called RAC protein. and that is important for actin polymerization. When polymerization is going to happen, then the actin molecule, they are going to extend themselves, and cell can actually enhance its size. You have CDC42. This is the molecule which actually controls the cell polarity, and then cell polarity and also directional sensing, which direction the cell needs to move. So this protein plays a very important role in this. You have RHOA, RHOA, and this protein plays very important role in actinomycin contraction. So cell will also require the contraction movement. So this is going to help in that, right? So if cell is moving from one place to the another one, the cytoskeleton rearrangement is going to happen. So if I show you with a kind of illustration, you will see the cell is going to move from one place, and it's going to change its shape, and that shape change is going to happen because of the actin cytoskeleton, and then it's going to further move by those change in the by the change in the cytoskeleton. So you can see over here actin cytoskeleton is changing its shape. So you have cytoskeleton called cytoskeleton rearrangement that is happening, so that cell can move from one place to another one, right? All right, let's understand vesicular changes, that how vesicular changes happens in case of inflammation. So you have a system called Kallikrein system, Kallikrein-Kinin system. Kallikrein-Kinin system is basically an important system for inflammation, where whenever there is injury happening, the release of a factor called factor 12 will happen. That factor 12 is also known as Hagman factor, and this factor actually is important in converting inactivated kallikrein, which is pre-kallikrein. Let me write it down, pre-kallikrein, and that is the inactivated form, into activated, which is kallikrein, right? So from pre-kallikrein, it's going to convert into kallikrein, and this kallikrein is nothing but a protease molecule. Protease molecule is going to act on a protein called kininogen. Kininogen is going to get converted into bradykinin. That is why this system is known as Kallikrein-Kinin system. So you have bradykinin, and this bradykinin is potent, really strong inflammatory peptide, highly strong inflammatory peptide. So let me write it down, inflammatory peptide, right? And this inflammatory peptide is important for specifically for leukocyte migration, for pain, for edema, all those things. This peptide is really important. Now what it does, actually it opens the endothelial junction. Next point is opening of endothelial junctions. Whenever bradykinin is acting, it's going to cause opening of endothelial junctions. And let's imagine we have blood vessels right over here. So I can show with the illustration. So let's imagine this is the blood vessel, and you have a lot of intercellular components inside that. And then what bradykinin is going to do, it's going to help moving these molecules to the site of infection, and that will also cause of edema, that swelling happens, and that is because of this particular pathway. So I hope now you understand this particular pathway nicely. So let's talk about mast cell activation and also release of histamine. This is really important, histamine release, to understand an inflammation, right? So just imagine you have a mast cell right over here. Whenever this mast cell is going to get activated, and the mast cell activation, if happens during using this particular pathway. So let's say this cell got activated, you have molecules like bradykinin. Okay, so this is one molecule, bradykinin. You have second molecule, which is complement. Okay, and then you can have another molecule which is allergens. So by using all these molecules, allergens, right? Allergens. So these molecules can cause activation of mast cells, and then that mast cell is going to accumulate a lot of calcium inside. So just imagine this is the next cell that I'm drawing as the activated mast cell. Inside that you have these green colored the molecules. These are high level of calcium. So high calcium level, when you have, basically there are granules that are present inside the mast cells. Small granules are there. Inside those granules you have molecules that are getting released, and it's called degranulation. This degranulation is induced by the high calcium level, and what it's going to cause is going to release the mediators, and that will cause vasodilation. You have vesicular permeability that is going to happen immediately. This reaction is going to happen. You have gaps that are going to enhance if there is any gap in the blood vessels. You widens the gaps, and then high edema is going to happen. So you have a lot of swelling in that region. So that is happening because of all these molecular pathways. They are playing an important role in inflammation, right? Okay, so now let's move on to the next part. All right, now let's talk about another cascade, which is arachidonic acid cascade. Really important pathway in inflammation, arachidonic acid cascade, right? So, and that is also linked with mast cells. So let's draw the mast cell right over here. And this is the whenever there is this activated mast cell, which we already understood how that got activated. So activated mast cell is going to release a molecule called phospholipase A2. And this is going to target the membranes. Let me write it down. Phospholipase A2. This enzyme is going to target the lipids in the membrane. So you have lipids, phospholipids that are there in the membrane. So these are the phospholipids I'm drawing, and these are the large membrane phospholipids that are there. And from there, when this enzyme is going to act, it's going to release from these phospholipids, it's going to release or form the molecule which is arachidonic acid. So by the activity of phospholipase A2, the arachidonic acid is going to get formed, right? And you have the COX-2 pathway. You have next is the COX-2 pathway, and in that case, what you have is arachidonic acid, right? The further processing of this arachidonic acid is going to be happening through COX-2 enzyme. COX-2 protein is going to convert arachidonic acid. So this is the substrate for this one. It's going to get converted into prostaglandins. Okay. And also prostaglandins, and you also have thromboxanes. So thromboxanes. So these molecules are going to get converted from arachidonic acid. And then let's talk about a little bit more about prostaglandins, because these are really important mediators, right? Prostaglandins, you have PGE2, which will cause vasodilation, and it's also fever inducing. It's going to cause vasodilation, it's going to cause fever, it's going to cause pain, right? Specifically targeting the sensory nerve ending. Because of activation of sensory nerve ending, you feel the pain. And then you also have PGF2A, okay? That is going to play an important role in vascular changes and further vascular changes, and that will cause smooth muscle contraction. So smooth muscle contraction is related with the PGF2A. Okay. Next, what you have is that we already discussed thromboxanes. You have, right? So thromboxane. Let me write it down. Thromboxane and A2. Specifically, it's also known as TKA2, right? And then it's going to cause platelet aggregation and also vasoconstriction. Platelet aggregation, we know what are platelets, and it's also going to cause vasoconstriction in this case, right? So thromboxane is playing an important role in case of functioning of the platelets aggregation and also vasoconstriction. Next is lipoxygenase pathway. So many pathways are there, right? So we are just discussing inflammation and different components of inflammation. So let's talk about lipoxygenase pathway. So you have the molecule called LTB4. So various cells, they're going to release this particular molecule. So let me create that illustration. So these are the molecules. These are the cells secreting this molecule, and these molecules, they play a very important role. So you have LTC4 and LTD4. These are going to cause the vesicular permeability. You also increase in vesicular permeability is going to happen because of this lipoxygenase pathway. And then further, you have bronchoconstriction. Second point, let me write it down, bronchoconstriction. Okay, bronchoconstriction is going to happen because of this. After lipoxygenase pathway, you have complement activation. There are complement proteins that are going to play an important role, right? So let me write it down, complement activation. Complement activation, you need to know what are those complement molecules. The first one is C3A, second one is C5A. Always remember these two molecules when it comes to inflammation and complement pathway. So you have these are also known as anaphylotoxin. So that means causing potent inflammation. So they will cause strong inflammation, strong inflammatory response. What is the function of these molecules? They can, let me write it down. Functions, right? So function number 1 is stimulation of the mast cells. Mast cells, they are going to get stimulated with these molecules. That's first one. Second, it's going to enhance the vesicular permeability. Okay, and next one, what it's going to do is going to recruit the leukocytes. White blood cells, they are going to come again to the site of infection. That we have already understood how complement plays an important role in attracting the immune cells, right? So this covers, most of the important roles of the molecules and pathways in case of inflammation, especially when it is related with biomolecular markers associated with inflammation. We talked about so many things. We talk about, if I summarize this, we talked about what is inflammation, about what are PAMPs, what are further we discussed about DAMPs. We also discussed about PRRs. Then we understood how TLR, they are going to activate the NF-κB, and how NF-κB is going to release the cytokines, and what are the function of all those cytokines. Further, we talked about endothelial activation, that how P-selectin and also E-selectin is going to play an important role in cellular attachment. And then we talk about adhesions, we talk about integrins, we talk about ICAM, VCAM, those adhesion molecules. Then we talk about diapedesis, and how the cells is going to move through very, very narrow spaces. We talked about the movement of the cells also, like cytoskeleton rearrangement is going to happen, and what are the proteins that are important in cytoskeleton rearrangement. Then we talked about very, very important kallikrein and kinin system. We talked about role of bradykinin, which is really, really important molecule. And then we talked about mast cell role. We talked about how the activation of the mast cell is going to happen. Further, we talked about arachidonic acid cascade. We talked about COX-2 pathway and prostaglandins, and also lipoxygenase, as well as complement function. This covers the entire inflammation topic. Okay, so now what you can have is a detailed summary of all those things, and hopefully this video is going to help you understand how these pathways they are linked, how what are the different components of the inflammation, and how they are related, right? All right, I'll meet you in the next one. Till then, take care everyone, and please study with interest, study hard, and enjoy your study. That's more important, right? Enjoy your study. All right, we'll meet in the next one. Take care.